Okay, good afternoon, everyone. Today, let's continue our study about the medical research methods. Today's topic is about brief introduction of cell signaling. So for today's class, there will be guidelines for what we are going to learn today. So today, we were going to learn the answers for the questions listed here. So we will going to talk about why do cells communicate. That means why cells need to communicate to other cells, why they need to do so. The second questions will be how are signals transmitted between cells? So once cell A decided to communicate with cell B, then how to do that? How are signals transmitted between cells? Then next, how are the signals transmitted from cell membrane into the cell interior? So how the signals outside the cells move into the cells. And then next, we will discuss about how are signals transmitted within a cell. When the message delivered into the cells, how those signals transmitted within the cell. And then after that, when the cells receive those messages, what do signals affect cell function? So today we will answer all these questions, but only the brief answers for the question, because this cell communication is very, very complicated. So it's not easy to understand them well. So only we have a brief introduction or brief answer for the following questions. And maybe later, after the future study, we can know all this process better. So let's start one by one. First, why do cells communicate? Why do cells need to communicate with each other? So here is a sentence describing cell communication. Cells communicate with each other using chemicals called signaling molecules. So when they talk to each other, you can understand that, talk to each other, they use their language as their signaling molecules. That's their language they talk. And then one cell needs to secrete these molecules out. So first, one cell needs to produce these chemicals, which is called signaling molecules. And then other cells need to receive those messages. They need to detect the presence of the signaling molecule. They need to use the receptors present on their surface to receive those signaling molecules. So once the signaling molecules is detected, the cells receive those messages, will make changes. They need to respond to those signals. So this is a brief introduction or overall summary about cell communication. First, one cell needs to secrete the signaling molecules. Then other cells need to receive those molecules through receptors. And then after they're receiving these molecules, they need to respond to this signal. So this is a brief summary about the whole process, cell communication. Then here shows two figures. The first one, as you can see, to the left, this is a cell A. You can call it a cell A. As you can see, this cell A secretes a lot of signaling molecules, which is a little white dots in the figure. And then a neighboring cell B receive those signaling molecules, and then later they will respond to the cells. The second figure is another process, as you can see, T cell and cancer cell. We have learned the cancer therapy in the previous topics. As we know, because there's cancer antigens present in the tumor cells. Then the cancer antigens have been processed by the antigen presenting cells, and then they can activate the T cells. Then the T cells receive those cancer message, and then they can migrate to the cancer tissue, and then they can respond to these cancer cells. As you can see here, they can destroy the cancer cells or kill the cancer cells. So all these are examples for cell communication or cell interaction. So some cells can release their signal, and then others can receive that. And then the cells receive those messages, need to respond to the signal. Here, for their T cell, they will kill the cancer cells. So this is the cell communication. So why do cells communicate? Why they need to talk to each other? That's because all the cells in a multiple cellular organism, as we know, inside our body, there are thousands of cells inside our body. So as there are multiple cellular organisms, all the cells inside our body must work together for survival. So the cells need to alert the rest of the body if they need something. For example, if you are under starvation, you don't eat food for a long time, so you have no nutrition inside your body, then your body the cells need to give the signal that you need to lower down your metabolite rate so that you can survive for starvation. So this is one example. The cells need to alert the rest of the body if they need something, or you are under some harsh condition, so that all the cells can work together to maintain survival for their host. And also, if our body are infected by, for example, infected by other organisms, so those immune cells need to give out the signals so that all types of immunocells help to clear their infection to maintain their healthy condition inside our body. So the main purpose for cell communication is that all cells try to work together for survival. So the multicellular organisms also had to build the efficient transport pathways. Sometimes the cells, for example, in the pancreatic, they need to communicate with other tissues, for example, to the muscle. That's a long way to go. So in this time, the pancreatic organ maybe it can secrete insulin, and then this insulin can pass through the blood vessels so that to their target cells, for example, the muscle, the liver, so that they can exhibit their function. So the multicellular organisms also had to build efficient transport pathways. For example, the blood vessels to deliver those cell signaling molecules so that they can communicate well with the cells far away. Here, there's a cartoon picture to describe about the cell communication. For example, the cells to the left, he said, let's decide an exact time to release some hormones. And then the cell nearby it maybe will respond to that. When would you like to do so? How about 10 minutes later? Then that's how cells work together to maintain the health of our body. So that's the reason why cell needs to communicate to each other. Then the second question, how are signals transmitted between cells? Once they want to talk to each other, how to communicate, how to do that? So here, there's one concept for that is cell signaling. So this concept, I hope you can understand. What is cell signaling? Cell signaling is about communication between different groups of cells and tissues. So one group of cells may tell the others or another group of cells what to do. As you can see here, this blue star represents the cellular signaling molecules, which is produced by one group of cells. And then this yellow or orange cells here, it's our target cells. So as you can see, the target cells have a green structure, which is a receptor. As these ligands or the signaling molecules bind to the receptor, and then those receptors can receive the message from other cells. They know what to do to respond to this signal. So this is the cell signaling, which means about the communication between different groups of cells or tissues. So the communication is completed by the form or by the interaction of ligand receptor. The ligand is the signaling molecules, and receptor is those most potent on the cell surface to receive those messages. So they need the ligand receptor binding or interaction to complete this cell communication. So here, let's imagine that cell A wants to send a message to cell B. So what cell A will do? Step 1, cell A secretes molecules that carry this message or signal. These molecules are known as the signaling molecules. As you can see in this figure, the signaling molecules secreted by cell A is represented for this red bubble. So this is the message produced by cell A. Then these molecules are called signaling molecules, and they function like a postman or messenger. So they carry the information for cell A to go to the cell B. And signaling molecules can be quite a lot of things. They could be liquids, proteins, or even gases. For example, the carbon oxide, or they can be the nitric oxide. Those are gases. So these signaling molecules can pass away in their extracellular matrix to reach cell B. So in their step 2, in order to receive the message, cell B needs a receptor. You can think this receptor like a specific mailbox to receive this signaling molecules. This receptor is a protein that can bind to signaling molecule. They are located in 2 ways. Some of them are located on the cell surface to bind the external molecules. Others, they can locate it inside the cells, which are the cytoplasmic receptors that can bind to the signaling molecules within the cells. So there's 2 types of receptors. One type located on the surface, as the figure here, located in the surface of the cells, so they can bind to the signaling molecules. But some of the signaling molecules can run inside the cells. So those molecules are bind to the cytoplasmic receptors inside the cell. So there's 2 types of receptor. No matter which time, you need receptors to receive these signaling molecules. So that's step 2. Then let's talk about the types of cell signaling. So cell signaling can be broadly divided into 2 types. First is the intracellular signaling. Another one is intercellular signaling, and these 2 are different. In many cases, signaling might involve cells talking to themselves and to others in order to generate a response. So for the intracellular signaling occurs within the cells in response to the internal and external stimuli. So we can receive the signal from inside or outside the cells, and then the cells in response to those stimuli, and then they can produce the intracellular signaling. Then, in other words, it is simply when a cell is talking to itself and working independently. That's intracellular signaling. On the other hand, another way, intercellular signaling is where a cell talks to the other cells of the body. So that's two major types: talking to themselves or talking to others. You can simply understand that. Okay, so this form shows the chemical signalings here. As you can see, there's 4 different types of the cell signaling. The first one is the cell target itself. As you can see, the cells can, So here, as you can see, our cells can produce their signaling molecules themselves, and then they receive those message by themselves. They talk to themselves. So this is a cell targets itself. It's called autocrine. Then the second type is that 2 cells are closely together and they are connected by the gap junction here. It's the which draw light tunnels there. That's a gap junction. Cell A can produce their cell signaling molecules and then receive by their cell B through these gap junctions. So this signaling across gap junctions. Then the next one is the cell targets a nearby cell. As you can see, those 2 cells are closely to each other, but they are not linked or connected by gap junctions. There's space between them, right? So then cell A can secrete some of the signaling molecules, and then those molecules will pass through the extracellular matrix and then reach to cell B. Then cell B can accept those messages from the receptors and then finish their message, the cell communication. So this time we called paracline cell signaling. And then the last one is the cell targets a distant cell through the brainstem. As you can see, there's 2 cells far away. So the signaling molecules cannot just migrate to the B cells, to the cell B. Then these molecules need to circulate in their blood vessel so that reach to the target cells, which has the specific receptors for those signaling molecules. And then they can bind together and then finish their message deliver. So as you can see, all these 4 figures, their signaling molecules look similar, right? They are all red bubbles or red balls. As we test, the sequence of the cell signaling remains the same, which means that although they deliver by 4 different types, however, those cellular signaling molecules are very similar. They are almost the same for most of the cells, but just different from the distance separating 2 communicating cells. So cell signaling can be divided based on the distance between these 2 groups of communicating cells. So this is types of cell signaling. Then we will talk for each type with more detail. The first one is the autocrime signaling. So sometimes cells can produce signaling molecules that bind to receptors on their own membrane. So in this way, it is possible for a cell to send message to themselves. So they talk to themselves. It sounds strange, right? Why the cells need to talk to themselves? Actually, it's very important for their cell function. The autocrine signaling is essential during special moment. For example, during the development, because they need to make sure correct cells division and maintenance of the cell identity. For example, one of the autocrine signaling molecules is this prostaglandin, which we call the PG. So this autocrine signaling molecule is involved in a lot of process, including the cell proliferation, differentiation, and cell apoptosis. This PG not only affect the neighboring cells, but mostly they can affect their own cells. So this is one kind of autocrime signaling molecules, the PG. So although this way is a little bit strange, but I think we can understand that. You can think of it as setting reminders and writing notes to yourself. It may seem weird, but it's necessary sometimes, right? That's make sure you don't forget things. You can respond to the correct date, the correct thing on time. So I think that makes sense, right? Okay, next one is the direct contact signaling. Some cells lie very close to each other. As you can see, these two cells are connected by the gapped junctions. So they are closely lie to each other, and they are directly contact. So such cells have passengers, which we also can call the terminals that connect them. So signaling molecules can easily pass through these passages. This feature enables a group of cells to respond to a signal received by just one cell. So you can consider this way of communication more private. They have their own contact information. The cell B and cell A are closely attached to each other. They have a link, which we call the passage, to link them, or you can call it the gap junction. So these terminals are private to themselves. All the message from cell A can pass directly or only to cell B. So this kind is direct contact signaling. So it's also common inside our body. For example, the regulation of interneuron excitability by gap junction. It's very commonly found in the neuron cells. So those excitability signal need to transfer correctly, and the direction needs to be make sure it's correct. Otherwise, we will have very severe side effect. So this direct contact signaling communication can be found for their interneuron excitability communication inside their central nerve system. So this is the second type, direct contact signaling. Then the last one, the next one is the Paracline Signaling. As you can see, these two cells are close to each other. However, they are not directly contact. There is some intercellular space between them, right? So cell A can secrete some of their signaling molecules into the extracellular matrix, then received by the cell B. So this form of communication takes place between cells that are near each other but are not connected. In this case, the cells talk through the diffusion of the chemical signaling molecules across short distance. So here give the example for this paracline signaling, which is the synaptic signaling between the neurons. As you can see, on top, this is the presynaptic cell. At the bottom is the postsynaptic cells. So as you can see that two cells are not directly connected by each other. They are near to each other, but not directly connected. So from the presynaptic cells, the neuron transmitters are synthesized and then released into their extracellular matrix. And then this neuron transmitter can diffuse in their extracellular spaces or matrix, and then received by the receptors on these postsynaptic cells, so that they can finish the communication between these two cells to function normally for the neurons. So this is the paracline signaling. Okay, the last type is endocrine signaling. So this is a method employed by cells that are far apart from each other. So they are far away from each other. Like a package that is shipped internationally, signaling molecules may travel through the bloodstream to reach the target cell. Such molecules are called hormones. Most of them are hormones. For example, the insulin, which is synthesized in the beta cells, then they don't function inside the beta cells. They need to transfer inside the blood vessels so that go to the distant target cells. For example, target to the muscle cells, target to the liver cells. So they need to travel through the bloodstream to reach the target cells. Most of them are hormones. That's the endocrine signaling. So we have introduced the concept of cell signaling, and then we introduced 4 types of cell signaling. And here, let's talk more about the importance of cell signaling. Communication among the many cells of the blood enable us to respond and adapt to the ever-changing environment. As we know, when animals or human beings living in the environment usually not, they are changing forever, right? For example, the temperature inside a day will change. In the morning, maybe cool. At noon, maybe hot. Maybe at night, it's cold. So our environment will be changing ever, all the time. So from a developmental aspect, cell signaling ensures that all body organs and tissues are on the same page. So we need to make sure that all the cells or all the body organs or tissues responds to the same changing environment. We need to make sure that once, for example, some of the cells know the change of the environment, they need to inform the other cells the changing. For example, if the hosts get infection by the virus, and then they are found by the immunocells first, and then these immunocells need to alert rest of the cells inside the body so that the whole body can prepare or work together to fight back for the infection. So that's the meaning or the importance for cell signaling. As I mentioned before, the cells need to work together for survival. That is the cell signaling can guarantees that things like cell functioning, size, location, and number are kept in check. In this way, cells of different specialized tissue maintain their identities and functions. For example, the cardiac cells are only present in the heart, and the hepatic cells are only found in the liver, and both perform their respective specialized role. So the cells need this cell signaling to respond to the changing environment. They need to communicate with each other to maintain their normal function and their location or size inside the body. Okay, that's the second question. How are signals transmitted between cells? They are transmitted through the cell signaling. There's 4 types. And then how are signalings transmitted across cell membranes into the cell interior? Okay, let's see. That's we finished step 1, step 2. The step 1 is cell A produce the signaling molecules, and then step 2, those molecules are recognized by the receptors on cell B, and then step 3, the binding of the molecule to its respective receptor on cell B can activate the receptor. So after the ligand receptor binding, the receptor will be activated. So this activation is brought about by a change in the shape of the receptor. So the binding will activate the receptor, and then the receptor will be changed in the shape. And the active receptor of cell B can now launch a series of events in which the message gets amplified and spread to other parts of the cells, and sometimes even other molecules. So this step is called transduction. And this process is called signal transduction pathway. What does that mean? So as I mentioned, step 1, cell A produce the signaling molecules here, and then they transfer, or they migrate, or they just deliver to cell B. So here there's a cell surface receptor on cell B. So these molecules bind to the receptor, and then step 3, this receptor after binding to the ligands, and then this receptor will be activated. So after activated, this receptor will change a little bit, sometimes change in shape, and then these receptors can pass this message get from outside the cells into the cells. Then this signaling will be amplified and spread to the other parts of the cells. Sometimes they can synthesize, or they can transfer this message in the form of secondary message. Then this step is called transduction. This process is called signaling transduction pathway. So this is the step 3. How to deliver the message from outside the cells into the cells. That's through this process, signal transduction pathway. So here shows the concept about that, signal transduction. Signal transduction refers to how the presence of an extracellular signal can produce a change in the intracellular state of cells without the initial signal crossing the membrane. As you can see here, the first message of the signaling molecules, they only stop, or they ended in the binding of receptor, right? So these signaling molecules do not directly go into the cells. On the other hand, or instead, this binding of the signaling molecule and receptor will change the receptor, and then the message can pass into the cells through the receptor. Through the receptor. That's signal transduction. They refers to extracellular signal produce a change inside the cell, and then they don't need the initial signal crossing the membrane. Then the next concept is the signal transduction pathway. So the transmission of the signal inside the cells is continued either by a series of biochemical changes within the cell, or by modification of the cell membrane potential by the movement of ions in or out the cell. What does that mean? So after the binding of ligand and receptor, the message is transmitted into the cell. By how? They can initiate or start a series of biochemical changes inside the cell, or they can change the cell membrane potential by changing their ion channels, so that they can deliver the message from outside the cells into the cells. So that's the signal transduction pathways. Okay. Here shows the signal transduction receptor. There's more information about the receptors. So the receptors are a group of proteins that can recognize the signaling molecules by binding to them. So there's some specificity of the receptors. First is the ligand specificity. That means the receptor, they will have their conformational structure of the receptor, so they can only bind to their specific ligands. So that's the ligand specificity, which means that special ligands recognized by their specific receptors, and then they can bind to each other, which are conformational fixed, lie a lock to the key. They are complementary fixed to each other. Then after the binding, the receptor will get the conformational change, and then they can get activated and then deliver their message from outside the cells into the cells. Then the second characteristic for the receptor is the binding specificity. So what does that mean? It means that different receptors can be found on the same cell, which means that the same cell, for example, the membranes of the cells, they can have different kinds of receptors. And on the other hand, the same receptor, for example, the toll-like receptor 4, can be found on different cells. The same receptor can be found in different cells. They can be found in dendritic cells. They can be found in cardiac cells. They can be found in the macrophage. So the same receptor, toll-like receptor 4, can be found in different cells. And the same cell, they can have different receptors. They can have the toll-like receptor 4. They can have the fibroblast growth factor receptor on the same cell. So this is the binding specificity. So the locational distribution of the receptors can be different. One cell can have different receptors. The same receptor can be found on different types of cells. That's the second characteristic, binding specificity of the receptor. And the last one is the effector specificity. So the binding of the ligands and receptors depend on their specificity structure. So that means that the ligand and receptor are complementary fixed to each other, so they depend on specific structure or conformational structure of the ligands and receptors. And then the effector specificity also depend on the types of cells. So different types of cells, they when they respond to the same signaling, they may have different effector. They have different response back to the same message in different cells. So that's the receptor specificity of the receptor. Okay. Next, let's talk about the types of signaling transducing receptors. Here shows 4 general classes of signal transducing receptors. The first one is receptors that penetrate the plasma membrane and have intrinsic enzymatic activity or are enzyme associated, which we call those receptors enzyme link receptors. We will talk about that later. Then the second time, I think a lot of students may be familiar with this time, the G protein coupled receptor. This kind of receptor have been introduced in a lot of classes, including the biochemistry, including the cellular biology. So they will all introduce these receptors because they play important roles in most of the physiology function of the cells. So those are receptors that are coupled inside the cell to the G protein. Okay, next is the receptors that are found intracellularly and on ligand binding directly alter gene transcription. Those receptors are nuclear receptors. The last one can control the ion in or out of the cell membrane. So they are called the ligand-gated ion channels. We will introduce that one by one. The first one are the receptors with intrinsic enzymatic activity. What does that mean? As you can see the photo or figure here, as you can see, this yellow one, yellow structure is the receptors with intrinsic enzymatic activity. As you can see, there are 3 different domains for these receptors. First, here, as the laser pen pointing, this is the domain on the extracellular matrix. So this is the outside of the cells. This yellow one is the cell membrane, and then this is on the top, on the bottom, and this is the intracellular space. So this as you can see, these receptors are transmembrane. They have the transmembrane structure. One part outside the cells, one part in the cell membrane, one part inside the cells. So 3 different domains for these cells. Okay, as you can see, there are domains outside the cells. They are the molecular binding domains, as you can see. When they recognize the molecules secreted by other cells, this is the signaling molecules, then this molecule can bind to this receptor. As you can see, after binding, the conformational structure of the cells change. At the very beginning, without the binding, these receptors are separated into 2 monomer, right? After the binding to the ligand or to the signaling molecule, then they form a third dimer. They bind to each other to form a third dimer. And then inside the cells, they have the intrinsic enzymatic activity, which means that inside the cells, this part, they can function like an enzyme. So what are enzymes? Enzyme is mostly proteins. Most of the enzymes are proteins, right? They can catalyze biochemistry reactions with very high rates, right? So inside the cells, after they receive the molecules, what do they do? As you can see, there's the protein tyrosine kinase, which at the very beginning are in the form of inactive form. So they are not functioning. They are inactive. And then after the receptor are formed into a dimer, after this ligand binding, they form the dimer. And then as you can see, this protein tyrosine kinase are phosphorylated, which means that they are attached to the phosphor group. That phosphor group is from ATP. So ATP transfer one of the phosphor group to this protein tyrosine kinase. So this kinase change from the inactive form into the active form. So they are now working. They can catalyze bioreactions. So as you can see, inside the cells, the substrate protein can be phosphorylated by this protein tyrosine kinase, and then they can get phosphorylated substrate protein. So this substrate protein will be phosphorylated. So for this, the whole reaction, after the binding of the receptor and the ligands, then the outside signaling, this molecular signaling, transfer through this receptor, then this receptor can activate the intrinsic enzymatic activity of the tyrosine, for example, the tyrosine kinase, and then they can phosphorylated the downstream substrate so that the outside signaling can transfer into the cells through this phosphorylation. This is the receptor with the intrinsic enzymatic activity. They can function as a receptor to receive the ligand, and then also they can function like an enzyme to phosphorylate the downstream substrate. So by doing so, they can transfer the information from outside the cells into the cells. So that's the receptors with intrinsic enzymatic activity. So here shows some more tyrosine kinase receptors, including the EGF receptor, the PDGF receptor, the nerve growth factor receptor, the insulin receptor, IGF-1 receptor. So a lot of receptors are functioning like this enzymatic, with this enzymatic activity. So this is the first time. Then the second time is the famous J protein coupled receptors, which we also called GPCR. So if you remember my second class, we talked about the drug target, right? If you remember, more than 50% of the targeted therapies are targeted to receptor. And among those targeted therapy to the receptors, most of them are targeted to this G protein coupled receptors. So why they are so popular drug targets for most of the research? That's because this G protein coupled receptors can bind to a lot of ligands, including the hormone, the light, the odorous molecules, the neurotransmitter, the neuropeptide, the chemotatic factors, and so on. They can bind to a lot of ligands. So target to this G protein coupled receptors can block a lot of different pathways so that they can be used for treatment of many diseases. So this is a very popular drug target for drug development, the G protein coupled receptors. So here, the researchers or the scientists have found many types of G proteins. Here shows some of them, GS, GI, and GO, and GT, and GPA1. And so on. So they located in different locations inside the cells. For example, the GS can be found in the liver, the adipose tissue, in the kidney, and the orient follicle. So they can be found widely or anywhere inside our body. And then the ligands for this signaling pathway could include the epinephrine, the glycogen, and other hormones. So after this stimuli bind to this G protein coupled receptor, then after the receptor are activated, they can activate the downstream effectors. For example, the GS protein can activate the downstream adenylyl cyclase, which can promote the synthesis of the CAMP, which is a very common secondary message inside the cells. So the effect, including activate the G-coupled proteins, including glucogen breakdown, the fat breakdown, the conservation of water, increase estrogen and progesterone synthesis. So as you can see, different types of G proteins, they located in different of the tissue or organs. After binding to different stimulus, and they can exhibit different functions inside the cells. That's the G-coupled proteins and their physiological effects. Okay, let's talk more about the diversity of the G protein coupled receptor and signaling transduction system. As you can see here, the G protein coupled receptor, which is look like this, as you can see. Also, they include the domain outside the cells located in the extracellular matrix. So they are the domain for the ligand binding. And also they have the transmembrane domains. As you can see, there's 7 transmembrane domains in the cell membrane. As you can count 1, 2, 3, 4, 5, 6, 7. There are 7 transmembrane domains. And then inside the cells are the coupled G protein coupled domain here. As you can see, this domain can couple to this G potent and then exhibit different or diverse functions inside the cells. This is the G protein coupled receptor, their structure. And the next type of receptor is the nuclear receptors. As you can find for the name, these receptors are located inside the cells. So they are called the nuclear receptors. Usually those nuclear receptors, their ligands include the steroid hormones, the thyroid hormones, the retinoids, or vitamin D. And this is the ligands for these nuclear receptors. So as you can see, this is the representative figures for the nuclear receptors. So as you can see, all of the receptors, they have a domain which are labeled in green in this figure. These are the DNA binding domain of the nuclear receptor. So as you can see, why these DNA binding domains are important? That's because they are closely related to the function of their nuclear receptors. So as you can see, the first figure here, the B, inactive receptor. So as you can see, this is the 3D figure for the nuclear receptor. As you can see, they have the ligand binding domain here, which are binding to the inhibitor proteins. So these inhibitory proteins can block the DNA binding domain so that they can close their DNA binding domains. So in this time, in this condition, these nuclear receptors are inactive. So when they bind to their specific ligands, such as the steroid hormones or serine hormones, and then, as you can see, the conformational structure of the receptor change a little bit, right? This time, the inhibitory receptor will dissociate, or they will lose from this receptor. Instead, the co-activator proteins will bind to maintain the active form of receptor. And also, this DNA binding domain will expose out, and then they can bind to their specific DNA so that they can promote or inhibit the transcription of the target genes. So this is the nuclear receptors, which is located inside the cells. Okay, next is the ion channel receptors. These ion channels, I think it's easy to understand. They can control the in or out of the ions from the membrane. So for the inactive form of this ion channel receptor, the terminal is closed, so the ions cannot run through this, run into or run out the cells. It's blocked. However, after this ion channel receptor bind to their receptors, the conformational structure of the receptors change. So the shape of the receptors change. So now the gate is opened, and then the ions can run through the terminal. So this is the ion channel receptor. They can bind to their receptors. So those ligands include acetylcholine and GABA. That's the common ligands for the ion channel receptors. Okay, this is the summary about the types of receptor, effectors, linkage. The first one is the ion channel receptors. After binding to their ligands, their terminals will open or close. Then the ions can run through the cell membrane and getting the depolarization or hyperpolarization, and then change the cellular effects. And the second type is the G protein coupled receptor. Those receptors, after binding to their ligands, and then they can activate the G protein and then produce the secondary messenger, and then change the cellular effects. And then the third type is direct control of the effector enzyme. So inside their cells, this receptor have the enzymatic activity. So after binding to the ligands, they can catalyze the bioreactions inside the cells, usually get some of their proteins be phosphorylated, and then change the cellular effects. And the last one, control of DNA transcription. They are the nuclear receptors. After binding to their ligands, they can expose their DNA binding region and then change the transcription of some of the genes and then make the cellular effects. So this is the types of receptors and effectors linkage. Okay, then the next question will be, how are signals transmitted within a cell? So then let's see this question. So the interaction between ligands and receptors causes the generation of second messengers. So here shows the examples of second messengers system, including the calcium ion, the CAMP, the CGMP, the nitric oxide, and the PLC, PKC, and the PIIP3 and DG. Those are the common second messengers inside our cells. So as the figure here, you can see the first messenger is produced by other cells. And then this first messenger bind to the receptors of cell B. And then they can activate the receptors. And then the receptors inside the cells can produce the second messenger. And this second messenger can exhibit their intracellular effects, help the cells to respond to the first messenger. So this is the second messenger system. So here shows some of the chemical formula of the second messengers, including this cyclic AMP. We also call that CAMP. So this AMP structure, I think everybody are familiar with that because the ATP, AMP, ADP, the structure I have learned in biochemistry class. So this is the pentose, this is the base, nitrogen base, and here attached to your phosphate group. Instead to form the linear phosphate group structure, it formed the cyclic structure here. So we call the cyclic AMP. This one is similar, just different from this nitrogen base. This is a G, not an A. So this CGMP. And here, another second messenger, that's the diacylglycerol. And then this is the IP3, and this is the calcium ion. These are all the common second messengers found in the cells. So the signal transduction at a glance. This is summary about the signal transduction. First, in the extracellular matrix, we have the signaling molecules from other cells. And then this outside signaling message can bind to the receptors on the target cells. Then these receptors have 4 types, right? Including the G-coupled protein receptors, the ion channel receptors, and the enzymatic linkage receptors, and the nuclear receptors, right? So these receptors can bind to their ligands and then respond to their message. They can either produce the second messengers inside the cells, or they can modify or change the biochemistry reactions inside the cells. For example, get some of the substrates phosphorylated and so on. So if they produce the second messengers, here shows the common second messengers inside the cells. And also this message can also activate the kinase inside the cells and then help to response inside the cells to the very first messengers outside the cells. This is the, how the cells communicate and feedback to their primary message from outside the cells. Okay, here shows the second messengers and their sources and their effect. First is the CAMP. They are produced by this enzyme, adenylyl cyclase. So the effect for the CAMP is to activate protein kinase. Another familiar messenger is the CGMP. It's produced by the guanine cyclase, and they can activate protein kinase. They can regulate ion channels. They can regulate phosphodiesterase esterase and calcium. They are produced by the ion channels in the endoplasmic reticulum and plasma membrane. And they can activate protein kinase, activate calcium modulated proteins, and so on. So each of the second messenger inside the cells can have their specific effect. After the produce of the second messengers, the cells will respond to the second messengers inside the cells and respond back to their very original first messengers outside the cells. So you can think this like first we receive the messengers from outside the cells by the receptor, and then the receptor can transfer those information into the second messenger inside the cells, and then the second messengers inside the cells can help the cells to feedback to the primary information. That's how the second messengers work. Okay, here is a case about the signal transduction pathway involving, for example, the nitric oxide and the cGMP that leads to vasodilation, the relax of their muscle cells. As you can see here, this one, the label pen pointing to this one, the first one, is endothelial cells. So the vessel cells, the endothelial cells. And then this endothelial cells receive their first message outside the cells, this acetylcholine, then bind to the receptor. So after the binding of the ligand and the receptor, they can produce the second messenger, which here is the calcium iron. This is the second message produced in the endothelial cells, responds to the first message, acetylcholine. And then this calcium iron can activate the nitric oxide synthase. Then they can produce the nitric oxide, the NO, which is one type of gas, right? Then this nitric oxide can diffuse from the endothelial cells to the neighboring smooth muscle cells. Then after binding to their receptor, then they can activate the guanine cyclase. Then they can synthesize another second messenger, which is the cGMP. And then this cGMP will promote the relaxation of the muscle cells, thus leading to the final vasodilation effect in the muscle cells. Okay, then last, how do signals affect cell function? So we already know why cells need to talk to each other, how they talk, how they receive the messenger, and then inside the cells, how they translate the outside messenger into the second messenger. And then last, how do signals affect the cell function? Then the step 4, the final step is response. The signaling molecule has now successfully delivered cell A's message to cell B. And then cell B must now decide how to respond to this message. The response will occur as a result of the signaling transduction pathway initiated by the activation of cell B's receptor. For example, if cell A has alert cell B to an increased level of blood sugar, cell A tells cell B there are too many sugar in the vessel, so they can result in the response that ramping up insulin production. We know that insulin is the only hormone can reduce the blood sugar level inside our body. So when cell A notices that too high level of glucose in blood, and then tell cell B, and then cell B can ramping up insulin production to respond to the high level of glucose in blood. That is step 4. After the communication, the target cells need to respond to the signal. They need to do something, right? Cell A tell that you need to do what, and then cell B get the message and then start working. That's the step 4, the final step, response. Okay, this is a case study about the whole cell communication, the glucogen signal transduction. As you can see here, the ligand, now the signaling molecule is the glucogen, which is the purple-blue rectangle here. Then after it binds to the specific receptor on the cell membrane. And then these receptors, you can see this is a G protein coupled receptor. So after the glucan bind to the receptor complex and then interacts with the G protein, and then ATP is converted to CAMP. So this gluconeogenic cyclase catalyze ATP to synthesize this CAMP. As we mentioned in the previous slide, this CAMP is one kind of second messenger, right? So this CAMP can activate the protein kinase A. Then this protein kinase A can activate its downstream phosphorylase B kinase. And then this kinase B can change from the inactive form to the active form by the catalyzation of protein kinase A. And then this phosphorylated phosphorylase B can activate more downstream kinase and then change the glucogen into glucose. So the glycogen can increase the glucose level in the blood. That's because they can promote the degradation of glycogen into glucose. And on the other hand, they can inactive the glucogen synthase kinase, so they can reduce the synthesis of the glycogen but promote the degradation of the glucogen, so that the total effect for the cells will increase the glucose level in their cell. So this is how the glucogen signal transduction first bind to the receptor, and then they can promote a series of bioreactions inside the cells, and then the cell will respond to the glucogen, increase the glucose concentration in the cells. So this is how the step 4 is performed, the response to the signal at the very beginning. Excuse me. Okay, again, here I would like to introduce one more example about the cell communication or cell interaction. So this is from my own lab, lab experiments from one of the project. So inside the body, we know that the high level of glucose can cause inflammation inside our body. This chronic inflammation can do harm to our heart. For example, it can cause the fibrosis, cause the hypertrophy, and the cardiac dysfunction inside the heart, right? And later, will deliver a specific disease, which is called DCM, diabetic cardiomyopathy. That's a heart injury caused by the diabetes. We all know that that's because the high level of glucose inside the blood or inside the heart cause inflammation and cause this injury, but we don't know how. How the glucose as a molecular signal, how to increase the inflammation, we don't know that. So raise this question, we want to design the experiment to find the answer. Okay, in our model, when the cells, for example, the cardiac cells, are challenged with the high concentration of glucose, we found that the following signaling pathway. First, this is a toll-like receptor, which is a receptor located on the surface of the cell, right? So this receptor can receive the glucose signaling and then pass this signaling inside the cells, which is through this MYD88, the MAPK and NF-kappa B pathway to induce inflammatory cytokine overexpressed. Then after the inflammatory cytokines increase, then cause the injury in the heart. However, we don't know if this glucose directly activate this toll-like receptor 4. We don't know if they function directly or indirectly. We don't know that. So based on that, we have several hypotheses. First, glucose, as we all know, they can be transferred by its transporter, the glucose GLUT. This is a glucose specific transporter for the glucose, deliver the glucose from outside the cells to inside the cells. So this is the, you can think, this is the car to deliver the glucose outside cells into the cells. Maybe the first hypothesis, maybe the glucose can transfer by this GLUT4 protein and then deliver into the cardiac cells and then cause the cardiac injury, the inflammation inside the cells. This is the first hypothesis. And the second hypothesis, maybe the glucose can directly bind to this toll-like receptor 4 and then cause the inflammatory signaling pathway downstream inside the cells. And also have another hypothesis, maybe the glucose working on the toll-like receptor 4 indirectly. We don't know which way is correct. So we design different experiments. First, we do the binding experiments with the toll-like receptor 4 and MD2 to the glucose. Then, as you can see here, we get a negative results, which means that glucose do not bind to toll-like receptor 4, and they do not bind to toll-like receptor 4's co-receptor MD2. They don't bind it. As you can see, the signal is negative. So glucose not directly due to these receptors. And then we perform another experiment. We use the technique to silence this GLUT4 protein, which means that we knock out this transporter inside the cells. As you can see, now the cells has no GLUT4, right? However, the high glucose still can activate the toll-like receptor 4 and MD2. As we can see, without the GLUT4, the binding of toll-like receptor 4 and MD2, which is induced by high glucose, are still working. So based on these two experiments, we know that the hypothesis 1, the glucose transfer by this GLUT4 not working, it's not work like this, not transport inside the cells and cause the injury, because without GLUT4 it's still working. And then we know that glucose do not directly bind to the toll-like receptor 4 and MD2, which means that they don't bind to each other directly. So the hypothesis 2 are still not working too. Then we know that maybe the glucose affect these receptors indirectly. So we further design experiments. We found that this is a very interesting experiment, as you can see here. The first, this white column means the control group. And then this gray and this red column means the high glucose challenge group. So the gray one, the glucose with the serum. So the cells, cardiac cells, culture in the medium with the serum. And then we add the high glucose concentration. As you can see, the inflammatory cytokines increase, right? The TNF alpha, the IL-6, those are both inflammatory cytokines. So the high glucose can induce inflammation inside the cells with the presence of the serum. However, if we remove the serum inside the culture system, you can see the inflammatory cytokine production is significantly reduced. So we get a conclusion that the medium, more specifically, the serum is required for the activation of this receptor when they challenge with high glucose. So they need serum. Then we repeat this experiment. I want to know what's the key component in the serum is required for this activation. And then we find that it's the fetal bone serum. It's needed for the experiment, which means that the proteins are needed for this system. As you can see, if I add this FBS, which is the bone serum, inside the system, the stimuli of the high glucose will work, right? So they need the proteins in the system. So why glucose activate the toll-like receptor 4 need protein? So the serum protein, what's the function for the serum protein to do that? Then we look through a lot of biochemistry books, and then we find that actually in vitro or in vivo, the glucose and the serum proteins can undergo an enzymatic reaction. So this reaction, we don't need enzyme to catalyze. The glucose plus the serum proteins, they can form the AGEs, which is the advanced glycation end products, the AGEs. So the AGEs, now we find that the AGEs also the high level of glucose, they both can activate this toll-like receptor 4 and MD2 to activate the downstream inflammatory pathway. So maybe it's not the glucose directly bind to this receptor, but the advanced glycation end products do so. So with this hypothesis, we design experiments. As you can see, now this time we don't use high level of glucose as the stimulator. We use the AGE instead as the stimulator. And then after adding the AGE, the advanced glycation end product for 15 minutes, you can see the activation of toll-like receptor 4 and MD2 because at the very beginning, no challenge or 5 challenge, the background for the toll-like receptor 4 binding to MD2. However, 15 minutes later, you can see the binding of toll-like receptor 4 to MD2 increase significantly, and then which means that the AGE, the advanced glycation end products, can activate the receptor conformational change. They can promote the binding of toll-like receptor 4 and MD2, which is a sign for the toll-like receptor 4 activation. And also, when we silence this MD2 or toll-like receptor 4, the high glucose or the AGEs will not form the complex together anymore. So we think maybe it's the advanced glycation end products as the cellular signaling molecule that bind to the receptor, toll-like receptor 4 and MD2. And so based on that, I designed more experiments to confirm that, including the Western blotting we introduced last class. We used the ELISA measurement and used the SPR and so on. We use a lot of techniques to confirm. Finally, we confirmed that yes, it's the advanced glycation end products directly bind to this receptor, toll-like receptor 4 and MD2. And then the binding of the toll-like receptor 4 MD2 can activate the downstream MYD88 and NF-kappa B. This NF-kappa B is one kind of the nuclear modulator, which can control a lot of inflammatory cytokine expression. So once the AGEs activate the toll-like receptor 4 activation, and then this NF-kappa B is activated inside the cells. So this AGEs can be, you can consider is the first messengers received outside the cells. And then this toll-like receptor 4 MD2 is the receptor they are specifically recognized by each other. And then this AGES can bind specifically to MD2 and then form the toll-like receptor dimer. And then this dimer has the enzyme link function inside the cells. They can promote the activation of downstream other enzymes, including the MYD88, the MAPK, and NF-kappa B. Then activate the gene transcription inside the cells. Then the cells responds to the AGE stimulates, and then they can produce a lot of intermediary cytokines, a lot, which is harmful to the cardiac cells, causing the cells cardiac fibrosis, causing the cell hypertrophy, and finally lead to the dysfunction of the heart, and then deliver the diabetic cardiac myopathy. So this is a case or an example for how the cellular signaling pathway study in the lab. We have the hypothesis and then design experiments using all of the experiment techniques to confirm our hypothesis and then get our conclusion. So what's the meaning for this basic research? So we know that it's the advanced glycation end products to promote the injury of the heart. And then we know that the receptor, toll-like receptor 4 and MD2 is needed for this pathological development of the disease. So we can use the inhibitor for the MD2, for example, which is called L6H21. It's an MD2 specific inhibitor which can block the function of MD2. So no matter how high level of AGES in the blood, when blocking the MD2, they cannot activate the downstream inflammatory injury, and then we can protect the heart from heart cardiac dysfunction. So this, when we study the whole signaling pathway, then we can find the appropriate or suitable drug target for drug development. So here we use the MD2 inhibitor as the potential candidate for the treatment of diabetic cardiac myopathy. So this is the example in our lab to study how to develop the diabetic cardiac myopathy and how to stop this disease. So this is the example for today's class. So I think that's all for today's class. I hope you enjoy the topic today and also can follow the whole study. Again, if you have any questions, welcome to send me email. And thanks again for your attention. Okay.
You're welcome, ma'am. Thank
you.
Thank you. Okay, bye-bye.
Goodbye.
Goodbye. Thank you.